So, are you able to hear me through the microphone? Okay. Thank you very much, Professor Hartov, and thank you for coming. I appreciate the opportunity to talk to you about the research program that we're doing at Hopkins, but also in more broad and general sense, the opportunities and challenges with delivery of heat using magnetic nanoparticles. And for much of the talk, I'll focus primarily on some of the general aspects of heat and energy, and then move on into some of the more specific things. And hopefully I will have enough time to end with some of the more, with at least one area of research that we've been dealing with that is still controversial, and hopefully that will motivate some discussion at the end. So some very simple concepts, but I think sometimes we forget these concepts, particularly when we work on developing tools for the clinic. And that is that, like any other drug or any drug development program, you really have three basic criteria that you have to meet for therapy. And that is delivery of the therapeutic agent. You have to worry about its distribution. Particularly, I mean, you want the distribution to be focused or targeted to the disease tissue and not elsewhere. And then, of course, you have to be able to deliver the right dose and measure this dose appropriately. So I think these three criteria are something that's typical for any drug development or any therapeutic development program. In this case, particularly, we're talking about energy. Energy is the therapeutic agent. And so that brings some unique challenges that does not exist with drugs. And more specifically, this energy is heat energy. And this brings up particular challenges both with delivery and with dosimetry, because unlike ionizing radiation for cancer therapy, heat energy is significantly different. So I'm going to step back a little bit, talk in general terms about the biological or the medical applications of heat and some of the motivation, and then work toward more specific aspects of this discussion or of this topic. So these are some data, cell culture data. PC3s are human prostate. It's a human prostate cancer cell line. And what these data show are survival curves. On the y-axis, you see percent of surviving fraction or percent survival assayed after a 1-hour exposure of the cells to these various temperatures, 37 degrees C, 40 degrees C, 42, 43, and so on, in a water bath. And the white bars correspond to just the heat. So at 37 degrees C, which is essentially the incubator temperature, this is their physiologic temperature, this is normal temperature. We don't measure a decrease in survival, and so that's good. And if we expose these or assault these cells with 5 gray of radiation, that's the red bar, orange bar, I guess, you see that survival is affected, and you end up with about 50, 60%, maybe 70% survival, and that's typical for most human cancer cells with about 5 gray of radiation. Not always true. Some are more sensitive than others, but that's a general ballpark number. You notice that as we increase the temperature, as we combine the radiation with higher temperature, we find something that's very interesting. And this is, in fact, the therapeutic. This is what generates the excitement for heat-based therapies. And that is, we can combine non-lethal heat, doses of heat energy, with low level or low cytotoxicity radiation and produce a significantly more cytotoxic effect or response. In other words, 42 degrees C by itself does not affect survival of the cell significantly, but when we combine it with the 5 gray of radiation, then the combination is greater than either alone. And that's what we're going after. And so there are clinical data that suggest this biologic observation is robust enough to translate into the clinic. And here are some clinical trial data of patients. I think This was cervical cancer. So it was a study on cervical cancer, and you see that not only is there an improved response, but there is improved survival. And so, of course, this is a significant motivator for developing heat-based therapies. In fact, Dartmouth has a history of this going back to the 70s. Dartmouth was a leader in probably still is, although the field has generally waned and is coming back, but Dartmouth in the 70s and 80s was a leader in heat-based therapies. So we also have the fact that heat-based therapies, or heat, does enhance chemotherapy. And so one can look in the literature, and one sees, if you look for what are the, look for evidence or a listing of the drugs that have an effect that is enhanced by heat, you find that many of the standard frontline therapies, doxorubicin, the platinum complexes, the Taxol complexes, these are all their effects are all enhanced by the addition of heat. So there is a significant motivator here from both biology and clinical data that's well published and well known to essentially get us to look very carefully at heat-based therapies and ways for improving them. And primarily in Europe, you find that there is an ongoing work, and in some cases it is becoming not quite standard of care, but certainly accepted practice to combine heat with either radiation or chemotherapeutic agents for specific cases of cancer in cervical, bladder, chest wall recurrences of breast cancer. And GBM is still, that's very much clinical trial stage as well as prostate. There are some challenges, and one of the primary challenges is really delivery. So why haven't heat-based therapies become more standard, or why are they not frontline standard of care? And one of the issues is, or one of the challenges with this is really delivery. So this, what I've shown you here, is a picture of, excuse me, it's an intraoperative intraperitoneal hyperthermia procedure in which the patient is opened up. It is an FDA, I think 2 years ago, this procedure received FDA approval for, it's a lavage with chemotherapeutic agent that is introduced in the peritoneal cavity at higher temperature, essentially for metastatic cancer of the peritoneum. Pretty, I don't know, it's crude, certainly a very invasive procedure, and personally I think we can do better. So the issue really becomes one of delivery, or at least one of the issues is delivery, and the fact that heat energy is mechanical incoherent energy that is indiscriminate. It does not discriminate between cancer tissue and normal tissue. And so its delivery is, or the delivery mechanisms, therefore we rely on technology to introduce methods of delivery that become specific to the cancer tissue. There is another issue, and that is the distribution. So as you know, I intend to talk about using magnetic nanoparticles for the heat-based delivery. And what I'm showing you here is essentially very crude pictures, very straightforward illustration of one of the issues. Here we have what you're looking at on the right-hand side are prostate cancer, human prostate cancer tumors that were grown in mice. We injected them directly with magnetic nanoparticle suspension, and that's the black area, and then took those out 3-4 hours later and sectioned the tumor. So on the right-hand side, does it look like we have a fairly good distribution of the nanoparticle therapeutic agent throughout the tumor? And the answer is absolutely not. So this was part of a study where we were working on methods, working to develop a method of delivering the particles directly into the tumor to try to maximize the intra-tumor distribution. Where the particles are, if they're the agent that delivers the heat, then obviously you want to have a good distribution of these particles in the tumor. This is a very obvious example. On the left-hand side, you can see that we clearly have succeeded in improving our distribution, but it's still not perfect. There are regions of the tumor that don't have particles, and I'll show you the consequences of that later. And then finally, there is the question of dosimetry, and for many in the audience, this is something that is well known. I certainly know Jack knows this. Jack Hoops knows this very well. So the idea is this. For human cells, the cytotoxic response to heat is dependent on time and temperature. Okay? So for a given temperature above a certain threshold, the exposure time determines the dose, and therefore there is a predictable response. If I am at 37 degrees C, which is physiologic temperature, obviously there is no hyperthermic dose because that is the normal condition. If I go to 40 degrees C and expose these cells for 1 or 2 or 3 hours, I really tend not to find any cytotoxic effect, and you can see that in the top left-hand curve and also in the bottom curve. You don't affect survival of human cells until you start to get to about 42.5 degrees C, at which point we see what's called a break point in the survival curve. So 42.5 degrees C for human cells represents that threshold where you start to see, at a reasonable amount of time of exposure, you start to see a break in the survival. And you can see this in the bottom left-hand corner on these malignant melanoma cells. At 42.5 degrees C, the survival curve starts to drop down. 43, it really drops down even more, and 44, and so on. For every 1 degree rise above this breakpoint temperature, we find that we double the rate of cell kill. Okay, so that has led to a formula. This is an Arrhenius relationship. That has led to this formula that one can find in the literature that is this cumulative equivalent minutes. It's a metric of the dose that captures or attempts to capture time at temperature for temperatures that are significantly above 43 degrees C. And I'm not talking about ablative temperatures or above 49 degrees C where you simply ablate or char the tissue. Okay, so those are essentially in 3 slides capturing the three challenges associated with this or any type of delivery, but specific with this. So the concept illustrated in a series of cartoons in one photo is to take magnetic nanoparticles that are suitably coated to be biocompatible and to permit effective delivery and effective distribution in the body. And for that, we consider antibody coating or attaching some other ligand that is able to bind to a molecular target on cancer cells. So, of course, we're looking for cancer-specific tags, if you will. And then, of course, after some period of time, the patient, or as in our case, many times it's the animals, are subjected then to an AC magnetic field, and the AC magnetic field causes a magnetic reversal, and through one of several mechanisms generates heat. It results in heat being generated by the particle, and now heat is deposited in the tissue where there's a sufficient concentration of the particles. So the equipment, and of course I talked a little bit about the radiation sensitization. So on the left-hand side, I'm just switching to the devices that we have at Johns Hopkins. In my lab, on the left-hand side, you see a picture, and for many of you, unless you've worked with Professor Hoops, it might not mean much to you, but we have an 80 kilowatt variable frequency induction power supply. That's what you see on the left-hand side. A couple of different coil geometries that we use. The one on the right here is one that we've actually been working to develop for easily 2 years, and finally we were able to take some really nice data with it a couple of weeks ago. Excuse me, that coil is one for high throughput cell culture, sort of plate geometry types of experiments. We really challenged engineering with that by having such an open, large gap sort of design with a homogeneous flux density in the working area. On the right-hand side, we have a small animal radiation research platform. I won't go into the details or the rationale for this, but those of you who have done work with small animals and in particular radiation research or radiation therapy with small animals, you know that there is a significant issue in using essentially a human clinical system slightly modified to treat mice or rats, and this SARP is designed to address that. It's an X-ray tube. We can do image-guided stereotactic beam delivery with a beam spot less than 1 millimeter in diameter, and you can see that right here on some film. So we'll go to talking a little bit about, in more detail, the combination of magnetic nanoparticles and AC magnetic fields for tumor-specific hyperthermia. And what we're really looking for is, number one, the combination of the magnetic particles with the AC magnetic fields provides us with an improved coupling of the energy source, or the energy, to the tissue-specific deposition that allows us to discriminate between tissues. And that's really the goal. If we have selective delivery, now our heat, our therapeutic agent, is delivered to the site, the intended site, and minimized, its delivery is minimized elsewhere. The nice thing about this is that we do have remote activation. Okay, so the particles are passive. They don't do anything until they're actually exposed to an AC magnetic field with the right field conditions, right field properties. So that does give the clinician some amount of control of the delivery of the therapeutic agent following intratumor or intravenous delivery, intravascular delivery of the nanoparticles. And that's actually a unique feature. And because these particles are imaging contrast agents, I won't get into that in too much, but we do have the potential to actually do some sort of dosimetry or dose calculation following some imaging, and by tuning the power, and you'll see some of these curves in a few minutes, we actually are able to, or the clinician can potentially control the dose rate or the total dose that's delivered. So I think that it has, at least conceptually, thermal therapy with magnetic nanoparticles and AC magnetic fields conceptually has a lot to offer. And so that's why I think there's a significant motivation for driving its development. Some of these diagrams, I have to thank Alan Foreman, who's in the audience, because he sweated these, I think, to make these. So I think I'll have to buy you a beer or something later, Alan. So this is just a slide to show you the general concept, the basic physics behind generating magnetic field. So if you have current flowing through a wire, through a loop, or through a solenoid, as you have here, you generate magnetic fields. And we induce magnetic fields by having current flowing through them. And so now another slide, courtesy of Alan Foreman, we can localize the energy to convert to heat with magnetic particles because of the unique interaction that AC magnetic fields have with materials. And there are basically a couple of modes of interaction. One is through hysteresis in magnetic materials that generates heat. Another one is through induction. But the two key features of the magnetic field that are important for this are really the amplitude and the frequency. And then, of course, there's a little spot here that talks about the different temperatures for the various modes of heat delivery, or interaction of heat with biological systems. For those of you who work with magnetic materials, you will recognize this loop here as an MH loop, and this is what I mean when I talk about magnetic hysteresis. In other words, if I expose a magnetic material to an external magnetic field, that external magnetic field will impose essentially a force, if you will, on the moment of the material and force it to respond by orienting itself to be in line with the magnetic field. And as I increase the magnitude of that external field, I force more of that material at its moment to orient itself with the magnetic field until I reach saturation, M sub S, magnetization saturation of the material. Now when I reverse that field, when I start to decrease the field and actually reverse its direction, the moment of my material doesn't necessarily retrace the same path, hence the hysteresis. And by essentially oscillating this magnetic field several times, I can get the material or the moment of the material to also respond in a similar manner, doesn't retrace its steps, and the area of that hysteresis loop is some indication or gives you some proportionality to how much energy is going to be released or delivered by the material, power loss, if you will. So a couple of slides on the magnetic nanoparticles that we developed. They're all iron oxide-based crystals of iron oxide. I will show you a little bit more about this particular particle that we've developed. The key feature here is actually on the right-hand side, top right-hand side. That's the power loss, the SAR, specific absorption rate of the particles in an AC magnetic field. In other words, how much power they deliver in watts per unit gram of iron. And the idea behind these particles is actually to get them to deliver more power at low field, because we want to minimize some of the other properties and both the engineering constraints and also enhance some of the safety features by using lower power fields for therapy. But they're crystalline iron oxide that are coated, and you can see here on the lower left-hand side on the graph that at different field strengths we actually are able to generate heat in a sample, and in about 40 or 50 seconds, if the right field conditions are applied, we get a change in temperature of about 30 or 40 degrees C. So we can actually generate a fair amount of heat fairly quickly with these particles. Another particle that we use, and also Professor Hoops uses in his laboratory, these particles are commercially available. These we've developed in several years ago when I was at another working in a company. These are now commercially available through Micromod particle technology in Germany. And these particles actually have some interesting properties in that they're able to produce a great deal of heat, but one has to activate them with a higher amplitude field. And we've done a fair amount of characterization of these particles. It is published, but these are essentially 100 nanometer particle with what we believe is a core comprising an aggregate of individual crystals, but the magnetic core is about 45 nanometers. So roughly, we tend to work in the 90 to 100 nanometer range for the particles. That's with a biocompatible polymer coating. It's either starch or dextran. Excuse me. So when we put different particle formulations in an AC magnetic field, they're all iron oxides. They're all dextran or starch iron oxides. For some reason, we don't see the same result. That's because the magnetic properties, even though chemically these particles are essentially the same, their magnetic properties are very different. The bottom curve, or the red data, the red line, represent Faradex. Faradex is an FDA-approved MR contrast agent. It does not heat in an AC magnetic field. These are all the same conditions for all of the particles. The blue curve are the particles that I just showed you earlier, the BNF particles. These particles produce a lot of heat. I can tell you, going up to about 480 or 500 watts per gram, 5 or 10 milligrams of this in a milliliter of water, the water boils in about 20, 30 seconds. So you can generate a great deal of heat and do a lot of interesting things with that, but you notice that these particles do not generate any heat down below about 20 kiloamps per meter. So we wanted to develop particles. That's one of the things we wanted to talk about, is delivering the right dose. We wanted to develop particles that are able to produce a reasonable amount of heat at these low fields, around 20 kiloamps per meter, because the SAR interaction, or the heat, the power loss of magnetic particles interacting with the magnetic field, scales with the frequency and the field strength. Okay? What that relationship is, you can see, is very complicated. It depends on your material. So that's why I put an x in the exponent there. In some cases, it's actually 0. Okay? Tissue, on the other hand, also, when one exposes a large region of tissue to an AC magnetic field, there is some interaction with the tissue. There is some power deposition, and its relationship scales with frequency squared, magnetic field amplitude, or power squared, and also the radius, to a first approximation, the radius of the volume of tissue that's exposed. It's not entirely true, but we'll just leave it at that for now. So the larger the portion of the patient who is exposed to a magnetic field, AC magnetic field of certain properties, the more nonspecific power deposition is going to be experienced by that patient, and that's manifest as a rise in temperature. And that's not what we want. So we really want to decrease the magnetic field or the exposure power, and that means that we want to increase the amount of power that we are able to generate from the particles for a given field. And so the lime green, or the green, the green trace is the power curve for particles that we developed since I arrived there, the JHU particles. And you can see that we did actually achieve, to some extent, we achieved the intended goal to develop particles, roughly 80 nanometers in diameter, to develop particles that are able to produce a reasonable amount of heating at low field. Notice that they saturate earlier than the BNF particles, and at saturation they don't produce as much heat. And so there's a lot that we could talk about on just the particles and the materials properties of these particles, but the shorthand is that the magnetic and the magnetocrystalline properties of these particles are very different, even though chemically they are the same, iron oxide. So if we take particles, in this case it was the JHU particles, and inject them into a tumor, in this case it was DU-145, which is a human prostate cancer cell line that we implanted into a nude mouse, a male nude mouse, grew the tumor, and we inserted a temperature probe into the tumor. We also measure the core body temperature of mice in all of our therapies, so that's the rectal temperature, because we really need to make sure that we maintain suitable differential heating. You can see that we are able to generate very nice hyperthermic temperatures at a given power and still maintain a very tolerable non-specific heat profile in the mouse. So this tumor, you would expect what, Jack? We would expect that. But that's not what we in fact saw. So these are tissue histology slides from 3 different tumors from the same study. On the left-hand side, we have tissue slices of DU-145 tumors that had been injected with saline. The mouse was put into the AMF, and what you find, this is H&E staining, hematoxylin eosin staining, and we see that the tissue looks very viable. Very nice, healthy cancer tissue, and that's invaded muscle, as you can see there. So I'm not a histologist, I'm not a pathologist, but this is what I'm told. Middle picture, we've taken a section of tumor where we injected the particles, and you can see, even under H&E staining, you can see where the particles are localized. They're very much localized in that brown gunky region. Those are the nanoparticles. Okay? We did not expose that tumor to the magnetic field. On the right-hand side, we have combined both the nanoparticle injection and the AMF to generate temperature profile, intratumor temperature profile, similar to the one that you've seen. And so what do you see? Where there were particles, you see evidence of coagulative necrosis, and a few cell diameters away, you don't see evidence that the cells have been significantly affected. So, yeah, that tumor, by all respects, should have behaved as if it was cooked, but what we instead found was that there was some growth delay, but the tumor regrew. So the intratumor distribution of the particles becomes a significant therapeutic factor in that you could actually measure a temperature that's sufficiently high for therapeutic effect. But if some region away you don't have a decent distribution of particles that are able to generate the same or this consistent dose that's cytotoxic or sufficiently cytotoxic, that cancer is going to regrow. So the challenges are the delivery, distribution, and I've so far only talked about the intratumor distribution and via one mode of delivery. And then, of course, the dose. And I don't think I'll have a lot of time to talk about dose and dosimetry. And furthermore, we don't have a lot of data on that yet because we're still developing the program. So at least when I talk to the clinicians at Hopkins, most of them would tell me that definitive disease or primary disease for most cancers, there are a few that are exceptions, is not a problem. So breast cancer, prostate cancer, the primary disease is not what they consider to be an issue. The issue is when the disease recurs, recurrent disease, widely disseminated disease, or metastatic disease. These are the issues for most cancers. So what do we do about that? Can we address that with this technology? Well, if we're going to think about that, then what are the specifications? What are the specs that our product would have to even have some hope of addressing this particular issue? Well, so a starting point, I've always thought, is maybe we start with, quote, smart particles that are able to, on their own, localize specifically to the cancer tissue or the target tissue. And that they are able to, when exposed to an AMF that has tolerable properties, that they're able to generate some sort of cytotoxic heat, roughly, let's say, something greater than 100 watts per gram of material. Now, I always assume that the uptake from some sort of targeted systemic delivery of targeted particles is going to be limited, let's say something on the order of 300 micrograms of therapeutic agent per gram of tumor. I think the device has to produce a consistent field, so a homogeneous field in the region of interest, however you define that region of interest to be. And I know in the discussions I've had with many of you, there are different schools of thought. This region of interest has to be small, this region of interest has to be big. But whatever that region of interest is, I think a consistent or homogeneous flux density field is imperative. Because for whatever particles you have, wherever they are, again, we're talking about widely disseminated disease or metastatic disease, you need to have a consistent dose rate, or you need to have a consistent production of power. And the only way you're going to get that, as you might remember from the curves that I showed you, is if you have a homogeneous flux density field to at least within some tolerable criteria. And then, of course, we also have to develop the methodology for appropriate dose and scheduling and dosimetry. And number 3 is probably one that goes much more is going to take place in the clinic, in clinical trials. So we've actually worked a fair bit on developing various targeted particles. And one here, these are electron micrographs. These are just kind of interesting pictures, I think, of particles that were targeted to the antigen, prostate-specific membrane antigen. It's not PSA. This is PSMA. It is overexpressed on most prostate cancer cells. It actually is also overexpressed in the vascular epithelium of just about all cancers. So it's kind of an interesting target. And here you see electron micrographs of PC3, prostate cancer cells, human prostate cancer cells that really, wild type, do not overexpress PSMA, but these guys are genetically modified to express the PSMA. And you see that these guys were exposed to 20 nanometer particles that were labeled with an anti-PSMA aptamer, in this case. And that cloud of material is really the nanoparticles. And then on the right-hand side, you have higher magnification. And really on the lower right-hand side, I thought this was cool. You can see that the cells are very actively taking up these particles. And that's actually part of the biology of PSMA. The PC3 variant that does not overexpress PSMA, which is a nice control, doesn't do this. So we don't see any of this particle localization. It's pretty quiet around these cell membranes. So that's kind of what we would like to do in vivo, in animals or in humans, with this kind of selective targeting. So I'm going to talk a little bit about some data or some work that we did several years ago that has been published, and also talk a little bit about some of the strategies that we're doing. So with regard to the prostate cancer and the PSMA, here in the cartoon I've got the antibody J591. It is a fully humanized antibody. It's in clinical trials for a variety of applications, including imaging. And so we're working to conjugate that to nanoparticles for this strategy. But the basic idea here is that we have iron oxide core or crystals that generate our therapeutic heating. And in this case, this cartoon is an iron oxide dextran matrix particle, SPEO particle, commercially available, because this is what we use for some of the data that I'm going to talk about, and that are already published. We coated that dextran with polyethylene glycol to enhance circulation time. And the selective targeting in the case that I'm going to talk about is a different antibody. And also we had put onto the antibody, we had put indium-111 as an imaging label to be able to characterize the biodistribution and blood clearance. So what you see in front of you are results, blood clearance on the left, organ distribution on the right, of these nanoparticles that were labeled with chimeric L6, an antibody that's humanized antibody for chimeric L6, or sorry, the L6 antigen, overexpressed in prostate and breast cancer. And these are female nude mice that were bearing HBT, human breast tumor 3477 xenografts. And we injected 2.5 milligrams of the particles that were labeled with the antibody and indium-111. And you can see that after 24 hours, we still had about 18 to 20% of the injected dose circulating in the blood. At least that's what we could tell. That's pretty good. On the right-hand side, the red bars, the first column is, or the first set of data are the tumor data in the tumor uptake data, and we get about 14% on average, 14% of the injected dose per gram of tumor is taken up, deposited into the tumor, which is actually quite good. But you notice something else. So we compared this, on the purple bars are the antibody alone with no nanoparticle, and then we also inactivated the antibody in another study and injected that. So we had the nanoparticle with the indium-111 label and the antibody, but the antibody was chemically inactivated, injected that into these mice to compare what the result is. And so what you see is the results of 3 sets of data across several organs. The purple, the antibody alone. The red, antibody nanoparticle construct, functional antibody nanoparticle construct. And the yellow, the antibody nanoparticle construct where the antibody is not functional. So what do you see? Well, when the antibody is not functional, where does the particle go? Mostly into the liver and the spleen. Does any stay in the tumor? Not really. Clearly, these particles get into the tumor, but they don't stay there. We also get some non-specific uptake in the lung, in the kidneys, brain, heart. That's driven by the antibody. So if you look at these, when I look at this, what I see is that the nanoparticle drives uptake in the liver and the spleen primarily. And the antibody non-specifically is deposited in the, well, there's some in the liver, in the lung, kidneys, brain, and other organs. And so when you combine the two, you get some sort of statistical average between these two. So I think this is really the challenge of delivery and distribution of any targeted nanoparticle formulation, is that you get rapid clearance from the blood, and that rapid clearance is driven by generally reticular endothelial system activity, macrophages in the lung and in the liver and the spleen that are actively sequestering these particles. And that, I think, is a significant challenge. So when we do electron micrographs in the tumor, or electron micrographs of section tumor, I should say, what we find is that we have evidence of nanoparticle localization to the cell membrane, tumor cell membranes. And when we compare tumors that were exposed to the AC magnetic field with sections of tumors that were not exposed to the AC magnetic field, on the right we have AC field exposure, on the left we don't have, you see evidence that there's basically some cytotoxic or localized ablative effect of having the nanoparticles combined with the AC magnetic field. So we do see biologic evidence of an effect. And then when in this case, in these papers that we published, we did look at tumor growth delay as the endpoint. We do see, and this is kind of a complicated bar graph, but on the far right, you see tumor growth measured as doubling, tripling, quadrupling time, quintupling time. On the y-axis on the left-hand side, you see the number of days to reach a particular benchmark in volume. And you see that on the far right, we have our controls, combined controls, that any of the treatment conditions that we had applied, we did see tumor growth delay. So the doubling time or tripling time was increased with our therapy compared to any of the controls. So we did have a sort of clinically relevant measure of effect. And so to summarize this portion of the talk, I guess one can say that there is a great deal of evidence that suggests that heat does have some clinical utility for, particularly as part of a multimodal therapeutic regimen for cancer with radiation and chemo. I think that it is possible with the right type of engineering on the particles and the right type of engineering on the device to achieve some sort of reasonable amount of targeted delivery of these types of constructs that will permit deposition of cytotoxic heat that could then be used as part of a therapeutic regimen. So, little debate, internal debate right now, whether to move on into this other topic. How much time do I have, Alex? Okay. All right. So, controversial topic. How small a tumor or cluster of cells could I conceivably treat with the magnetic nanoparticles? This idea, magnetic nanoparticle hyperthermia as a cancer therapy, is about 60 years old. In 1979, an American surgeon by the name of Robert Gordon proposed that having the particles inside the cancer cells would actually would give you a therapeutic advantage than particles outside or external hyperthermia, because the cell membrane can act as an insulator, trap some of the heat in, and help you generate a thermal gradient, temperature gradient inside the cell that's cytotoxic. And that actually drove, for about 10 or 20 years, it drove some of the research in this area. And in fact, there is a company in Germany now that is running clinical trials using magnetic nanoparticles that were developed around this sort of strategy. In the late 90s, early 2000s, a few mechanical engineers primarily started to question this notion and said, wait a minute, what about heat transfer and heat transfer dynamics? Where is this heat going to go? How can you actually have a nanometer, 100 nanometer diameter little particle acting as a source of heat in this massive sea of a cell cytoplasm, right? 10 microns in diameter, and actually generate any heat. So they started to question this, and there were some models that came out that said that no, you actually can't, you cannot kill a single cell with intracellular or any kind of nanoparticle, targeted or not, targeted nanoparticle or intracellular nanoparticle hyperthermia with current technology. So a postdoc and a resident of mine decided to test that, because apparently nobody has actually tested this experimentally. So here's the experiment. Take the nanoparticles, these are BNF nanoparticles, coat them with PDL, poly-D-lysine, which is a common transfecting agent, and this just enhances the uptake of the nanoparticles, which are actually by design not really taken up by the cells. Take those cells, and here's the critical part of the experiment. Transfect the cells, trypsinize the cells, or essentially lift them, wash them very carefully to make sure you remove all the extra particles that have not gone into the cells, and now you count the cells, and you centrifuge them all at precisely the same sort of rate so that you pack the cells into a pellet with a consistent packing density. Okay? And so I want to stress this point because it's going to become important later on. Now, if I vary the cell number, I can vary the size of this cell pellet, okay? But they all have the same consistent density. So basically what I'm doing is I'm deliberately changing the size of a particular collection of cells, all packed to the same density, and I can go down to a few cells or, several million cells. And so I can do this sort of hyperthermia test. And just to give you some idea, on the right-hand side, we've got Prussian blue staining electron slide picture of light microscope picture of Prussian blue stained cells. So you can see that these guys have taken up a lot of particles. And the heating curve we have on the top left-hand side, and the vertical and the horizontal dashed lines tell you what power we were working at. So we were basically working at the saturation point of these particles, roughly 480 watts per gram of iron, about the maximum that these particles can produce. Where are these particles? Electron micrographs tell us that, which is typical. These particles are internalized by the cells and packed into these endosomes. One of the criticisms of the theoretical models was that these engineers don't know anything about biology, and they just assume a homogeneous distribution of nanoparticles in the cells. Well, anybody who's done this experiment knows that that's not the case. Okay, so that was actually my criticism of at least one of the criticisms. Well, particles aren't homogeneously distributed, so it can't be right. And so now, just to not belabor the point, but basically the experiment, so you can go to a basic heat transfer model where you have a sphere that is generating or producing heat embedded or submerged in a medium having heat transfer product, let's say water, that acts as a heat sink. And you can, from this very sort of simple model, you can generate or calculate what the predicted temperature at the surface of that sphere ought to be, and what the temperature at the center of that sphere ought to be. And you can also see that these are the relationships on the left-hand side, top left-hand side, and D is the diameter of the sphere. So you can see that the temperature gradient, if you will, depends on the diameter squared, and P, which is the power. Okay? And the power here, because it's being generated by nanoparticles, is really just the concentration of the particles, product of the concentration of the particles, and the power loss factor of the concentration normalized specific power loss. And so here on the right-hand side, I just show you a graph. So what Mohammed and Owen did was they packed these cells into spheres. They put the suspension into the AC magnetic field coil, and they placed a temperature probe right at the cell pellet surface. And they measure the temperature rise. And what you see on the right-hand side is actually a cell pellet surface temperature when they turn the power on. So they can clearly are able to see a significant change in the temperature. So when we compare these measured temperatures with the predicted temperature from the model, this is what you get. Okay, so on first glance, and when I saw this, I'm like, okay, for me anyways, that's the end of the discussion. The model is able to reasonably well predict what kind of temperatures you're going to see in these various cell pellets. So what are the different conditions? Well, we've just varied the concentration of the nanoparticles in the cell. So the blue, I guess the dark blue, we have 199, roughly 200 picograms of iron. So these guys are loaded to the gills with particles. And then the green, we've got about 100 picograms of iron per cell, red 70, and then the light blue is 5 picograms of iron. And on the x-axis here we varied, or they varied, the cell diameter by varying the number of cells, same packing density. Okay? So the fact that the model and the data agree so well to me is actually quite interesting. Now, what was one of the key assumptions in this model? That the sphere is internally thermally transparent. So in other words, I don't have cells with cell membranes that are acting as insulators. So that model prediction would have broken down here. So the cells really don't, their cell membranes really don't seem to act as, at least from what we can tell, measurable thermal barriers. The other thing that you can actually sort of get from this, the corollary of this, is that you need to actually have a collection of cells, or for a given SAR of particles and given concentration of particles per cell, one has to amass a large enough collection of cells together to be able to generate a cytotoxic thermal gradient in that mass. So turn it another way, what's the smallest tumor you could conceivably treat with this technology? Well, best case, the very best case, which is 200 picograms of iron per cell, that is a lot. Okay, 200 picograms of iron per cell. Now I used to look at my survival curve. I've got to be somewhere around, let's say 300,000 cells. 200 to 500,000 cells. Roughly a millimeter. So that's the smallest tumor I could conceivably treat with this alone. Okay, now if I add radiation or chemo here, all of this shifts to the left. Right? I can go down to smaller cell numbers. Anyways, that is a bit of a controversial point, because for about 20 years, it seems, people wanted to believe that nanoparticle intracellular or cell membrane-bound hyperthermia is somehow better or more effective because it's localized to the cell. It doesn't seem to be the case. It seems that because the cell is predominantly water, it's in an aqueous medium, heat that is generated by these particles moves through the system and is dominated, its transfer is dominated by macroscopic heat transfer water. And so that is the conclusion that I would come to from those data. Anyway, so to finish up, just to acknowledge people in the group and other collaborators. Within radiation oncology, have a couple of postdocs. One of them has recently left, David Bordelon. He got a job in South Carolina. So I'm down to one postdoc. I have one resident, Owen Thomas, radiation oncology resident, MD, PhD, physical chemistry. That's why I think he is quite good. It's because of the physical chemistry background. A few summer students have helped out in this collaboration with JHU School of Public Health for the mass spectrometry. That's how we characterize our iron concentrations. And then a collaboration with NIST for the physical characterization and magnetic characterization of the particles, and an independent guy, electrical engineer, who, nothing in here, but he helps out with any of the electrical engineering, and then, of course, the funding sources. So thank you very much for your attention. Yes, I think for three reasons. One, the history of cancer therapy, I think, should teach us one thing, that there is no magic bullet. No one therapy is likely to succeed, I think. Second, all of the clinical data with heat-based or hyperthermia-based therapies tells us that heat alone does not generate a significant response, or certainly no durable response that is better than radiation or chemo. But when combined with those, significant improvement and significant response. And then third, I think some of these challenges are overwhelming if you look at this approach as a standalone agent.
When you say radiation, is that localized microwave heating?
No, that I would localized microwave heating I would count in the heat-based therapy camp, because that's non-ionizing radiation. When I talk about radiation, I mean ionizing radiation, X-ray, gamma.
Based on the same kind of analysis that you're presenting, one can assume that the brain-sized tumor, there'll be a shell on the outside that will simply just not reach any significant temperature.
It's possible that delta T at the surface is that calculation for what that surface temperature ought to be and that surface gradient ought to be. So basically, if I want a therapeutic threshold, I'm going to shoot for a delta T of about 5 or 6 degrees C. So assuming 37 degrees C is my baseline, and let's say 43 is my beginning for therapeutic effect, then I want to look for a delta T of 6 degrees Celsius. So actually, I'm able to generate a delta T. There was another graph that I didn't show up here. I'm able to generate delta T at the surface of 6 degrees and also in excess, but I just have to go to larger diameter particles. It's a surface to volume problem, really. It's heat loss at a surface relative to the volume of material that's generating that heat. Instead of the one of the
leukemia, a non-solid might be easier to establish efficacy. It's true we have sites where the spleen, bone marrow, and so forth may be hard to get to. Solid tumors are so much more difficult.
So I think, yes, the solid tumors do present some challenges. I think also that, well, so at least from the data we've taken and the heat transfer model analysis and so on, you do need a collection of cells that is somehow associated closely with one another, that reaches a certain critical size given particle concentration and power loss to be able to have some efficacy. Now, whether, what stage of tumor, the type of tumor, how solid that tumor, I think those are actually valid points. And I think in some cases, some solid tumors may be resistant to any sort of penetration by these particles. But on the other hand, I would say a well-vascularized solid tumor might actually be the perfect candidate for this kind of therapy. Whereas a leukemia, particularly individual cells or small, loose collections of cells, I think, at least the data that that I just presented at the end, I'm not hopeful that that's viable. I don't know.
I was just wondering, your prostate is solid tumor, and you mentioned that it's vascularized, but I'm sure you've thought about this. Vasculature is going to actually help carry a lot of heat. So where does that feature into this?
Yeah, that's a very good question. So I would respond in the following way. First of all, I think the typical notion of tumor vasculature and the tumor physiology is that the vasculature and the flow is a little bit more chaotic. And compared to normal tissue, perfusion is not as efficient, and therefore heat transfer is expected to be rather less efficient. Okay? However, having said all of that, it is actually possible to generate sufficient heat, even with that, from by using antibody-targeted nanoparticles, I mean, we've demonstrated that, as other groups have demonstrated, that you actually are able to generate some heating. But yet another reason why one would want to consider this as part of a multimodal strategy, where yes, you generate some heat that sensitizes that region to a chemo or radiation therapeutic agent, so that you can actually improve your therapeutic ratio.
Right, but the core of the tumor may not be very well diffused, but the periphery is an aperture vessel. And, and that may be where it may be an issue, because it's not so difficult to debulk the core of the tumor. It's difficult is at the edges where it's a little more mature. But, yeah.
Yeah, I agree. I mean, and then one can actually turn that argument around and say, well, because it's so well vascularized, you're going to get more particles there, and as long as you have enough of the protein marker, a high enough expression, you're going to get more material there, and therefore you should be able to get more bang out of the heating. So I agree. I mean, these are all sort of nuances, and you really don't know until you actually get there, which is, of course, one of the reasons why the particle development programs drive the heating, are driven to produce particles with higher heating efficiency, so that for every unit mass of material that's in the tumor, you're actually generating more power. Oh. Yes. Yes, it does pose a potential problem. That's actually a manuscript that we just sent out, but I didn't have time to show those data. Very quickly, since you asked, we did a study where we injected a large volume or a large amount of non-targeted particles intravenously into mice, not bearing tumors. And then I think we waited 2 days, maybe 3 days, and then subjected these mice to an AC magnetic field. We also looked in the liver and the spleen, and did some iron characterization and histology and so on. And yes, it is possible to generate significant damage in liver and spleen by exposing a patient or a mouse to a combination of a high dose of particle with a high enough field to generate a significant amount of power. Yeah, you can damage significantly the liver and the spleen. So that is a concern going forward. I think that is one of the challenges that goes into this distribution category. So yes. Thank you.